Hello, my name is Victor Zberstis, and I'm on the team working behind the scenes on worldcommunitygrid.org, which has been harvesting spare processing time from computers, tablets, and smartphones around the world since 2004, putting these computing devices to good use while they would otherwise be consuming electrical power just waiting for that next keystroke or mouse movement. One of the research projects on World Community Grid is the Nutritious Rice for the World project from the University of Washington in Seattle, Washington. Please let me introduce Dr. Ling Hong Hung, who will give you an overview and an update of the progress of their research.
Thanks, Victor. So I'll be talking about the Nutritious Rice for the World project. This is where World Community Grid members took DNA sequences of rice genes, 62,000 in total, and predicted 10 billion final protein structures. First thing I want to give is an overview of where the talk is going to go. I'm going to talk about the science behind the project and basically why we did Nutritious Rice for the World. Then go through what World Community Grid accomplished, and then summarize the results. So, why Nutritious Rice for the World? Now, let's start off some of the ideas behind it and what we were trying to accomplish. The idea was, could we use the sequence of rice DNA or genes to make better strains of rice? Can we do this by using rice DNA sequences to assign biochemical functions to the corresponding genes? Now, typically, this type of process called gene annotation is done by comparing sequences of the genes to sequences where we know the function. This slide shows a typical search that might be done, where we would input a sequence of a new gene and search it against a database of other genes where we know the function. And so if a gene is similar in sequence to another gene, then it's likely to be evolutionarily related, and then we can surmise that it probably shares a similar function. However, the problem with rice genes is that they're very, very different from DNA sequences that we know. So we couldn't just do a simple sequence versus sequence comparison for many of these. So we did do this in some earlier work, but we did find that many of these DNA sequences were too different to pull out anything that was similar in sequence, and we couldn't find function that way. So now what? As I said, sequence comparison works through the principle that genes are closely related, have had little time to diverge, and probably functionally related as well. So it's an indirect method of inferring function. However, in nature, function is not carried out by the DNA itself, but by the proteins encoded by the DNA. So another method for determining the function of a gene is to compare the structure of the protein made by the gene against a database of proteins where you know the structure and you know the function. So that was the basic Nutritious Rice for the World strategy. The problem with protein structures is that if you want to determine them experimentally, this is a very, very difficult process. It takes months and years. And roughly 25,000 distinct protein folds have been deposited in the database. And but structure prediction, it can be done computationally, and it's much faster. And World Community Grid predicted 62,000 genes in just a couple of years. So I'm going to go into some of the details of protein structure prediction methodologies, how it's done, and why we did the way that we did do it. The easiest way and most accurate way to make a model of a protein is if you have a sequence and you use a sequence to find the sequence of a known structure. It's similar enough. Something like Rosetta will do this. This is very accurate, but it requires something we don't have, which is similarity in sequence to something which we already know. Another way of doing it is through molecular dynamics, something like folding at home. But for this to work, really the current molecular dynamics methods are really only accurate if you have a good accurate starting confirmation. Again, this is something we don't have, so we can't use this. So what we did use was de novo modeling, starting from new, using a piece of software called ProtinfoAB. It's not nearly as accurate, but it can make models without sequence similar to known proteins. And so that's what we did use. Okay, so let's take a look at the heart of the Nutritious Rice for the World project. What did the members and volunteers do with the cycles that they donated? The client program that was used is ProtinfoAB. It uses an energy function. It's not a real energy function. It's a statistical energy function that's derived from the properties of observed in known proteins. And then the the member computers searched for conformations that minimized energy. We need many, many structures because there are many, many false structures or bad structures that will have low, good energies. So you have to make many, many structures to get the best ones. So 62,000 genes were modeled and 10 billion structures were generated by the volunteers. Okay, I'm just going to go a little bit into what the program actually does in a little more detail. The best way that we've found to predict the structure of a protein is using a method called fragment assembly. What you do is you take small pieces of structure, of known structures, and you put them together to make your test conformations. The advantage of this is that you know that those fragments are going to have good protein-like local geometry, and so you've already reduced the conformation states that you need to search. Now, what we actually use, ProtinfoAB, is a variation of this. Instead of using actual fragments from proteins, the library of the collection of fragments are converted first to probability distribution functions. So the angles which are randomly obtained from these functions will mimic the distribution that is found in nature. And so this allows us to obtain angles that are the same distribution as angles from real proteins, but without actually restricting ourselves to the exact angles that are found in real proteins. So this allows us to search a little bit more broadly than we would if we were just using real fragments, which is an important thing because we are looking at very diverse sequences and probably a focus material a little bit more diverse than what normally we would deal with. So ProtinfoAB generates a series of structures, and it looks at these structures and it's trying to find a conformation that minimizes the energy. So if you go along the pathway, you'll generally get structures which are lower in energy. It chooses those. Once in a while, it chooses one which is slightly worse in order to sample the space. It gets stuck in a local pocket. Only the final structure in the trajectory of 10,000 conformations was retained. So while we generate 10 billion structures, and that's what we saved, over 100 trillion structures were actually generated during the project. Now, even 10 billion structures is a lot, and it's too many. So we did some post-processing to find the most accurate structures for each gene. We tried to get down to 5 best structures for each gene, which reduces this down to about 300,000 structures instead of 10 billion structures. The methods that we had used previously just don't work on this scale. We tried choosing the best structures using better energy functions, and this really didn't work that well. The best method we found was to look for agreement among different models in terms of structure, and this was done through a mathematical method called clustering. And during the course of the post-processing of the data, we developed some new, more accurate protein clustering methods that were fast enough and could handle the large data sets that we encountered in the Nutritious Rice for the World project. And this was a useful advance in protein structure prediction. And we also had to develop, again, because of the size of the data sets, we had to speed things up a little. And we developed some parallel GPU multi-core friendly methods and algorithms, which, again, though they're not part of the community part of the project, it was part of the post-process part of the data. These are still very useful methods, protein structure prediction in general. So we've now made all these models available for download. The entire set of 10 billion models can be downloaded upon request. That's 4 terabytes, so it's not up there on the website. But you can download all 300,000 best models, or you can search for, or researchers can search for a specific gene or set of genes and download just those models. One of the things is that there's different sets of nomenclature for the rice genes. So we set up a database to cross-reference this so that you can enter any one of these names and pull out the corresponding rice models. And this slide shows the different names and models that can be pulled out. You can also pull out multiple domains for a single gene, as we modeled multiple domains, not just the genes. Some of the genes were fairly large, and we wound up breaking them into smaller domains that we could actually model. So how is this going to be used, or how can it be used? This is a schematic of a biochemical pathway. This is a KEGG pathway. KEGG is a set of annotated and conserved biochemical pathways. And this is a plant pathway. So it's basically a biochemical circuit diagram, with the rectangles being proteins and the circles being the products actually made by the proteins. The red lines in this diagram show physical or predicted physical interactions between the components of proteins. And by looking at what the annotation information will help biologists is determine which rice proteins fit into the components in these pathways and allow them a better understanding of these pathways as they apply to rice. So this is a summary of what we did. So researchers can now use predicted structure to do structural comparisons and better understand the functions of these newly sequenced rice genes. During the process, we also developed some new, more accurate clustering techniques, which are very useful in protein folding simulations, not just the structure prediction methods which I've talked about, but also for molecular dynamics. And this is the basis of a bioinformatics paper that just came out. We have also developed some fast, parallel, and open source technologies for protein folding as a result of this project having analyzed the large amounts of data. And these are also the subject of several papers. The database results and any updates will be maintained at the original ProtinfoAB Nutritious Rice for the World webpage. And also, since I've left the group, I'm also going to be setting up a compilesoft.com site, which will also mirror these results. I'd like to, of course, thank all the people that were involved in this. This has been a very rewarding experience for myself, and it's been a real honor and pleasure to work with everyone involved. Ram Samadrala is the principal investigator for the project. He's the one who came up with the basic idea and provided the facilities and fundings and brought all the people together at UW for this project. Mike Gherkin, Mike Shannon, H.R. Dang, these are the sysadmins that without which we could not have handled that much data. Mike Gherkin helped a lot with the original data storage compression schemes, strategies to manage data, and did a lot of work interfacing with the IBM people in the early meetings. Mike Shannon recovered a large proportion of data which might have been lost. We didn't lose anything because of him. HOI helped with the post-processing, made sure that it didn't affect all the other things that we were doing on the cluster. I'd like to thank the IBM team who are listed there. Victor's especially, who's been there since the start of the project. Bill, who's now retired. Teddy, Jawan, all the others. These are the people that I regularly meet with and who've pushed me to complete this project. And most of all, this would have been completely impossible without the goodwill, enthusiasm, and interest of the volunteers of World Community Grid. And really, I thank you for this experience. It's been wonderful. And on that note, I'll end.
Well, thank you, Dr. Hung, for this informative talk about the Nutritious Rice for the World project and how our idle computer time through worldcommunitygrid.org has helped with this project. We hope those listening are contributing and will encourage their friends to also contribute their spare computing power by joining worldcommunitygrid.org. We have time for a few questions. First question is, how many varieties of rice genome were processed using World Community Grid?
There were 2.
What are the kinds of rice taken for this research work?
Japonica and Indica, so the Indian and Japanese versions of rice. And
how many genes are there in a typical rice genome compared to a human genome?
Well, I believe presently there are 50,000 genes that have been identified, protein coding regions. In contrast, it's roughly 20,000 protein coding genes in humans, so there are a lot more in rice.
How are scientists using your results to figure out which genes cause the best nutrition, drought resistance, and pest resistance, and so forth?
Well, that depends on our collaborators and people who are interested in the structures. The structures, as I said, can be used to search against a database of structures in order to try to get more information about the function. In the majority of cases where the sequence comparison will yield nothing, it can be used to help design mutations, to do experiments. I think really the idea is it's a resource for the real research, which is going to understand how these proteins interact in the biochemical pathways and how those biochemical pathways produce drug resistance, pest resistance, and more nutritious rice.
How long would you expect it to take for your research to lead to a safe new breed of improved rice? In other words, when do you think we'll see rice being grown based on this research?
Again, that really depends on our collaborators, but this is not easy work. The computation stuff is probably the easiest part of the entire process. The actual crop science, the biochemistry, the genetics, that's all very, very difficult and arduous work. So it's going to take a while.
Okay. Are your techniques applicable to other food crops or plants?
They should be. Again, this is a general problem with plant genomes, is that they are very, very different from, the sequences are very, very different from the mammalian genomes, which we're more familiar with, where we have a lot more data on. So the idea of simulating structures and using those structures in order to establish function is generally applicable. It's just finding the right number or an adequate number of cycles in order to do the calculations.
Well, this particular project is finished doing its computation on World Community Grid, and you've made the results publicly available to everyone on your websites there. Will you be also collaborating with other researchers on the further steps of this research?
I think Ron will be continuing with that. I myself probably will not.
Okay, and we have one more question. And somebody was asking who Ram is again.
Ram Samadrala, he's the principal investigator for the project at the University of Washington, Department of Microbiology.
Okay, well, thanks again, Dr. Hung, and those attending. Sorry if we didn't get to your question. Please use our forums at World Community Grid to post any additional questions. This concludes our webcast.